So my video is going to be on respiration, and the first step of cellular respiration is glycolysis, and basically glycolysis is converting glucose into two pyruvates. So then we start off with glucose, and it's broken down in half and converted into two PGAL molecules, and then after that PGL turns into NAD plus is used, turned it into two PGAP molecules, and then it's phosphorylated with ADP with a return of ATP, and then you get two PGA molecules. Then from that it turns into PP molecule, and then from that it's phosphorylated again with ADP, and you get one ATP for each PEP molecule. And after that, once it's phosphorylated, you get two pyruvates. And that gives you, what happens in glycolysis is you get a net gain of two ATPs and two NADH used in the electron transport system. And so glycolysis is basically you get a glucose molecule, two ADPs, and two NAD pluses, and then what you come out of from glycolysis is two pyruvates, four ATPs, and two NADH. Okay, so now in the Krebs cycle, first before we get into the Krebs cycle, the glycolysis, you get pyruvate, and then pyruvate goes into the mitochondria, and then there's a prep step where the pyruvate combines with a coenzyme A to produce acetyl-CoA, and then also NAD plus is used to make NADH, and then you get a byproduct of CO2 and NADH. So once you get the acetyl-CoA, it goes into the Krebs cycle, and it combines with a oxaloacetate to create citrate. The citrate then goes through a series of intermediates where NAD plus is used, and then we get a product of CO2 and NADH, and that happens through two intermediates. Then we get to this third intermediate, and we get ATP, and then it goes again, and then NAD plus is used in this intermediate to get NADH, and you get two of those, and then we go again, and then we use phosphorylation again to get NADH, and then we get again another molecule of oxaloacetate, and then this is used again in the Krebs cycle for acetyl-CoA. So after the Krebs cycle, we have the electron transport system. And what happens in the electron transport system is that NADH and FAD2 and FADH2 are used in order to create a proton pump that pumps hydrogen onto the outer membrane. So high energy electrons are used to pick up the hydrogen protons, and pump them to the outer space, and then they're picked on by electron acceptors and moved into the next protein system. And then again, hydrogen is pumped out, and then another electron acceptor is used and taken to the other protein system. And then the electron again pumps this hydrogen proton to the outside, and then ATP synthase is used, and these hydrogen protons are used in order to phosphorylate ADP to get ATP, and you get a net yield of 34 ATPs, and this is where we get the majority of our ATPs. Okay, so this is cellular respiration, and they both aerobic and anaerobic go through glycolysis, and for when oxygen is present, we go through the Krebs cycle electron transport chain, and we have a net yield of 34 ATP, and when oxygen is not present, we go through lactic acid and alcohol fermentation, which creates a total of 2 ATPs. So what happens in lactic acid fermentation is that we go through glycolysis, and we get the two pyruvates, And then pyruvates is then used with NADH to make lactate, and we get a byproduct of two NAD pluses that go again and are used in glycolysis. And what we get is we get the phosphorylation of ADP to ATP, which gives us our net gain of 2 ATPs.